Thank you, Andrew, for the invitation, and thank you, everyone, for coming. It's early, so that's always an achievement. How do you move? Okay. So Andrew has been talking about sliding windows, which is a very popular approach. The most popular approach is they are easy to understand, which is a benefit. But they are not lack of issues, as he mentioned before. One of them is how to choose the width of the window. Also, there is an intrinsic limitation in the resolution of sliding windows, because typically you need them long enough, and you might miss quick changes. And most importantly, and that's where I like to focus, they make a not completely efficient use of the data, because sliding windows, by definition, look at whatever is in the window, ignoring the rest of the data set. So that obviously makes means that changes between windows might be due to just statistical noise, just having an imperfect estimation. So you need to be very careful when assessing this change, because it might be due to just noise. So there's been some authors proposing tests and rigorous approaches to test for that. And some other authors have just claimed that, you know what, at least in the resting state, at least in fMRI, this is just hopeless. So there's no way to find dynamics. So my first message of my talk is that these problems, of which this inefficiency is one of them, but that's the one I want to focus on. These are not a fundamental problem. This is a technical problem, and therefore there must be an engineering solution about it. So that's what I'm going to try to talk about. So our approach is in this case to move from the sliding window approach to a state-based approach, where we assume that we can describe data using a discrete number of states, and then the data set gets represented by visits to these states. So in this case, every color is a different state, and in this case, I'm representing them using function connectivity matrices, but that's not necessarily always the case, so we have different representations of states, but I will talk about that later. So the critical point is that assuming, again, that we can reasonably describe the data set using a discrete number of states, this model, which is the hidden Markov model, gives you three things. Gives you an estimation of when the state happened, so what is the probability of each state happening at each time point. It gives you a description of what the states are, which are the parameters of these states, and that might contain information about function connectivity, or might contain information about the spectral properties of different things, and transition probability matrix, which tells you which is the probability of going from one state to another, because the HMM is a dynamical system, and these dynamics get parameterized by these probabilities. So again, under the assumption that you can reasonably describe the data using a discrete number of states, the inference of the HMM gives you when the state happened, which are the parameters of the states, and which is the probability from going from one state to another. So the benefits of this is that we don't need to specify the window length, although we need to specify the number of states, in principle. We can access fastest time scales, because the visits to the states can be quick, typically might be quicker than the typical choice of a sliding window. And most importantly, they make an efficient use of the data, which leads me to my message number 2, which is that differences between states are in the data. And let me exemplify this with an example. So we used a lot the data from the HCP. So in particular, the release that we normally use has 820 subjects, 1 hour data each. So that means that if we estimate 12 states, for each state, we have almost 70 hours of data to estimate the parameters of that state, as opposed to a typical choice of a sliding window, which is 1 minute. So I hope that comparison is talks by itself. So I've been vague about what a state is. A state is basically a mathematical object. It's a probability distribution, because the HMM is a probabilistic model. And most of my work in the last years have been about designing types of modalities of state descriptions that tailor different types of data, different data modalities. So I've been working on different types of HMM for EEG, MEG, or fMRI, or LFP. So that then comes message number 3, which is important to know the HMM or the state distributions are not biophysical models. So this is important. Therefore, the question about whether or not there are discrete states or how many states there are fair questions, are valid questions, are interesting questions, but they are less interesting than whether or not this offers an useful perspective of the data. So, in different words, does it help you to answer the question that you have? That is the important thing, to me, in my opinion. So the corollary is that this is one way of describing the data among others. ICA is another way. Autoregressive models is another way. Rafael is going to talk about that later. And this state-based approach, of which HMM is one of possibility, Joanna is going to talk about another one, is one possible way to describe the data. That doesn't mean that this is the ground truth. So I'm going to try to demonstrate that this is useful through a couple of questions that we were interested in and we investigated using this type of models. The first one, we use fMRI data. So the state distribution that we use for this is a Gaussian distribution. Basically contains, each state contains the mean activation and some pattern of function connectivity using a covariance matrix. And the question in this case is, we were interested in the temporal dynamics of the resting state networks. So people have been investigating and studying during many years which are the spatial properties of the different resting state networks, but we don't know that much about the dynamics of these networks. So if you remember, one of the elements of the HMM is the transition probability matrix, probability of going from one state to another. So the first thing we noticed here is that some transitions are just much more likely than others. Some transitions between networks happen very often, and some others just almost never happen. So we plot this as a graph where each node is a state and each arrow is a transition, where the thickness of the arrow is how probable is that transition. The second thing we notice is that states or networks cluster into two groups that we came to name metastates, such that the probability of going from one state to another within a metastate is much higher than the probability of going from one state to another across metastates. So that means that there's a temporal hierarchy, a slower temporal hierarchy in the networks. If you look at the spatial characteristics of these metastates, so in this case, I'm looking at the connectivity of each region with the rest of the brain, you see that one of these big Uber networks or metastates is focusing mostly about sensory motor regions, visual, auditory, whereas the other is what we will understand by higher cognition, DMN, language, extensive prefrontal. So there is a clear spatial separation between these two metastates also in the spatially. So we can relate this to behavior because the HCP comes with a large number of phenotypes and psychological information, and we found a relationship between how much time you spend in the metastate and some of these psychological traits. It's not a super strong relationship, but it's quite significant. What is stronger is the fact that these metastates are very subject-specific, so are very robust across sessions. So people tend to just stick to that number of to that metastate profile across sessions, and also they are very heritable. So you tend to have a similar metastate profile than members of your family, and that relationship is stronger if this is your twin, and is even stronger if it's your identical twin. So that was what I want to talk about MEG. I'm going to talk about MEG, about fMRI. I'm now going to talk about MEG now. So basically, we are interested in the spectral properties of the different networks, something that we don't have that much using fMRI, obviously. We've been trying different things over the years to try to characterize these spectral resting-state networks. The first one, even before I came to Oxford, was done by some colleagues. We used a sort of similar approach that I used for fMRI on the power envelopes of the signal. So basically, they bandpassed the signal, they focus on some frequency of interest, and they computed the power, and then run some HMM on that, such that its states get characterized by the activation in that band in terms of power and power covariations, called function connectivity in terms of power correlations. The issue here is obviously, by definition, we don't know anything about phase. We lose everything about phase, and phase is something that I think we all agree this is an interesting thing. So attempt number 2, I thought, well, let's characterize its state using a multivariate autoregressive model, which contains a lot of information about the frequency and contains information about phase. So that was an okay idea, and it works really well for a few regions. So when the channels have rich spectral information, like in LFP or even in MEG, but it doesn't work that well when you want to model the whole brain. So basically, I don't want to go into details, but it overfits the data when you want to model, say, 40 channels of your brain. This is because the autoregressive model is a very powerful model, even though it's linear. Again, Rafael will talk about that later. So attempt number 3, it was a proposed recently simplification of the autoregressive model, which basically accounts for cross correlations in the data and loses a few things. Loses, for example, directionality between channels. So we lose a few things, but in exchange, we get over this issue of overfitting, and we can find resting-state networks that are spectrally defined. In particular, they are characterized by a certain pattern of power, a certain pattern of phase coupling. So here the questions are there consistent phase coupling networks in the brain? And one network that we're interested in is the DMN. How is the DMN in terms of phase coupling, and which are its spectral features? So I'm going to start with question number 1. So we run this on a number of resting-state subjects, 50 subjects, and we estimate a number of states. I'm going to show you a couple of them here. One is a sensory motor state, one is a visual state. Both get characterized, as I say, by power and phase coupling, and these networks are spectrally defined. So these quantities are defined as a function of frequency. We use some data-driven approach to factorize this frequency information into 3 components that we named slow, alpha, and beta. They are not what you canonically will understand as alpha and beta, but as I say, this is a data-driven approach, so it's what the data says. And we find, as you would expect, sensory motor network, it seems to be more active in beta, is well known, and visual is mostly about alpha. So this is not very surprising. So we found another 2 states that were more interesting to us. We called them anterior higher order cognitive and posterior higher order cognitive. The anterior one has a lot of activity in the prefrontal regions and temporal poles, and very, very interestingly, and we are very excited about this because if you work in MEG, you know the PCC is a tough guy. So we found a strong, really strong phase coupling between the PCC and the prefrontal regions, which is, for us, was quite interesting, again, in very slow frequencies. Whereas the posterior higher order also has PCC involved. PCC is key in fMRI, but in MEG is difficult, as I say. And this time, the PCC is interacting with posterior regions, basically parietal regions. So question number 2, what is the DMN here? The answer is that there is no such thing, but if you overlap these 2 states, it really looks like the DMN. So we verified that quantitatively using some meta-analysis, and we found that what is commonly understood by a DMN in fMRI here seems to decouple into 2 components that happen at different time points, and also they are spectrally different. So they happen at different frequencies. One is slow, one the other is alpha. So I want to end up with some conclusions. Again, the HMM is a handy, not a biophysical model, it's a handy way of describing the data, might be useful for some questions, and it provides a well description of dynamics that you can then relate to behavior in different ways. So we use it here to investigate temporal dynamics of resting-state networks in fMRI and some spectral characteristics of the networks in MEG, focusing on the DMN. I have a second slide of conclusion with my 4 messages. I'm not going to repeat them because I don't want to be annoying. I don't have too much time, but I'm going to leave them for my last 10 seconds. Well, not even 10. Thank you very much.